Ruth, for the introduction. And thank you to the SAW board and organizing committee for inviting me out. It's always nice to come out west. I was just saying, I haven't been to Regina since my friend was actually at an RCM depot maybe a decade ago, so it's neat to be back here. So yeah, my research group, our focus of research is environmental toxicology, which is basically a fancy way of looking at the impact of contaminants on the environment. And just a little bit of background, these are some of the organisms, I like to call them our tools, for understanding the impacts. So these are some of the species that we culture in our lab. Some of them are freshwater, some of them marine, and some of them terrestrial. So our research is quite diverse in terms of the ecosystems we study and the organisms that we study. The newest member of the group is the purple sea urchin there in the middle. So it's been fun learning how to keep them alive and to be able to use them in testing. So I'm going to talk about some of the work that we've done looking at the impact of microplastics on some of these organisms. As you're probably aware, microplastics is sort of this emerging contaminant. I'm sure you've seen some very sensational headlines: microplastics in lungs, microplastics in, the most recent one was the microplastics in bottled water and that sort of thing. So the big issue right now is, we know they're there, but what's the impact on human health, but then also the ecosystems. So just a little bit of background about plastic. This was actually really interesting when I started to look into it. Around World War II, chemical era, production of plastics, we started to discover different types of plastics, and production started to accelerate. And because they have these properties that are very useful, they're light, they're strong, flexible, versatile, cheap to make. So you can make a variety of products out of them. And there are positives to plastics. I think sometimes they get a bad rap. They have revolutionized a number of industries like transportation, the medical industry, because again, they're easy to sterilize. You can make them into a variety of different products. So they have really helped in advancing a number of industries. Again, helmets, incubators, water purification. But then the dark side is that the convenience, they provide a convenience, they're cheap to make, but that convenience leads to this concept of sort of that throwaway culture, right? We use it once and then get rid of it. It's not that expensive. We can just get more, right? And these are some of the original advertisements from the 40s and the 50s, Tupperware, Tupperware parties and that sort of thing. So this is really interesting. Half the plastics ever made were made in the last 15 years. Which is kind of crazy to think about. It just gives you an idea of how production has scaled in the last couple decades. So 2.3 million tons were manufactured in the 1950, and then 2015, that's a little dated, it's gone up to 448 million tons. And it's expected to double from that 448 million tons. It's expected to double by 2050. So that's production of new plastic. That's not considering any recycling. So we're producing huge amounts of new plastic on a regular basis, and it's climbing quickly. So there's roughly about 8 million tons of plastic is released into the ocean each year. And the issue is that they're relatively long life in the environment, right? And again, it depends on the polymer, it depends on the plastic, but it can be anywhere from 5 to 400 years. That's the half-life, so for half of it to break down. So that's the problem. I mean, these are the properties we want in the material, but the problem is when it releases into the environment, we also have to deal with the consequences of that. So, I mean, it is a global problem. They have had meetings to discuss, what are we going to do about it? So world agrees that plastic is a waste crisis, but again, trying to figure out the solution to it is much more challenging. This is an iconic, and again, here I'm talking about macroplastics, not microplastics yet, but this is an iconic image. This is an albatross chick. So the parents forage on the ocean, bring the food back to the chicks. And so this was a chick found in a remote atoll, the Kura Atoll, and it had died. And when they did a necropsy, you can see these are all the different products that were found in it, right? So the parents were picking it up and then feeding that to the offspring. And again, this is a macroplastic issue. And you all may have heard about the giant garbage patch in the Pacific Ocean. Due to prevailing currents, all the plastic that goes into the Pacific ends up in this huge patch. And 99% of the garbage patch is plastic. And it gives you an idea of the size of it, right? Like you see Hawaii down here in the corner, San Francisco on the coast, gives you a sense of how big this garbage patch is. So we've got an issue with macroplastic, but one of the concerns is that plastic will break down, which is a good thing, but it breaks down to the tiny little particles that then continue to persist in the environment called microplastics. And the definition of a microplastic is anything less than 5 millimeters in size. And microplastics can be all kinds of different products, but one of the things we've sort of discovered is that you can find microplastics virtually anywhere. So the deepest parts of the ocean, right, the abyssal plains, the Arctic, the Antarctic, I mean, they're everywhere. So they travel through ocean currents, but then also lighter particles can be carried, by atmospheric deposition. So there's no question they're everywhere. And even in us, right, you'll find them. I mean, a human consumes about 1 credit card a year in microplastics, which is kind of weird to think about, but that's what scientists have sort of discovered. And so the big question is they're everywhere, they're in us, but what's the adverse, are they going to have an adverse effect on health and ecosystems? So where are they coming from? I mean, you can imagine there's all kinds of different particles or products: tires, synthetic textiles, marine coatings, road markings, personal care products, plastic pellets. And these are just some pictures, right? You can see these, our tires wear over time, so the tiny little particles are released. Over here are in red, those are red microfibers from a fleece blanket. So when you wash your fleece blanket and the washing machine, it gives off those. And then up above is a personal care product like shampoo or a facial scrub, where they put tiny little microplastics in it to add as an abrasive, right? So you get that, I guess, deep cleaning, on your facial scrubs and that sort of thing. Now, those they've in certain jurisdictions they've outlawed those, so you just can't put microplastics in personal care products anymore. It's just not seen as a useful, right? The benefit doesn't outweigh the cost. Another interesting thing, this is one thing that my research group has found, is that actually tiny little invertebrates, they'll actually make microplastics. So this is an isopod or a pill bug or roly-poly or woodlouse, however, there's different terms, but they will actually chew on the plastic. If you have plastic, that's been disposed in the forest and the field, they'll actually, they're eating the bacteria and stuff that's growing on the surface of the plastic, but they'll actually inadvertently create microplastics. So that's sort of interesting. So for freshwater ecosystems, a lot of the focus on plastics and microplastics has been on oceans. And more research is being done now in freshwater ecosystems. And a big source of microplastic to freshwater ecosystems are wastewater treatment plants. And that's true for a lot of contaminants, because wastewater treatment plants, they receive wastewater from all kinds of different sources. And so microplastics have been found to be released by them. So we did a study in the Grand River watershed. The Grand River watershed is in Ontario. It's the largest watershed in southern Ontario. There's a number of municipalities or large wastewater treatment plants throughout the watershed. And now the water quality is drastically improved as wastewater treatment has improved. That's been clear in the Grand River. But we're interested in what the microplastic situation is. So we've focused on one group of organisms, freshwater mussels. And these are our native mussels. So these aren't zebras or quagga. This is sort of our native species that we have in Canada. And the reason we chose them is they're sedentary. They don't really move much. And they filter massive quantities of water on a daily basis. So their potential exposure to microplastics would be pretty high. And they don't move. So, when we're looking at different populations, we kind of have some idea of whatever they ingested came from upstream from that location. So we looked at this species in particular, and the red dots is where we sampled mussels. And then the blue dots are the location of wastewater treatment facilities. So you can see that we've sampled really far upstream where there's less probably exposure, and then further downstream, around Kitchener, Waterloo, Cambridge, the Tri-City area. I mean, there's hundreds of thousands of people that are those wastewater treatment plants are servicing. And so what we do, a little gruesome, maybe there should be a warning, but I guess if you go to the restaurant and eat mussels, you're not worried about this. But what we do is we open up the mussels. We've sampled from the different locations. We digest away all the organic material, the tissue and everything else, and then we filter it to isolate the microplastics, because the microplastics don't break down. I mean, so that's a positive when you're trying to sort of find them. Everything else will break down and they'll be left over. And then we look at them underneath the microscope to sort of identify them, measure them. We look at, are they a fiber? Are they a particle? Are they a film? We look at these sort of things. We look at color. But one of the things we need to do too is confirm that they're actually plastic. Because there's a lot, as you can imagine, there's a lot of things a mussel's going to ingest that maybe we didn't digest, right? Tiny little stone, tiny shard of glass, stuff over time. And so we have to confirm it's actually plastic. So we use this Raman spectroscopy. I'm not going to get into it. Basically, we shoot it with a laser, and then it tells us, if it's actually a polymer. Is it polyethylene, polypropylene, polystyrene? And so that's one, to confirm it's a plastic. But the other thing is, ultimately we want to figure out where these plastics are coming from that we're finding inside the organism. So part of that is looking at, are they fibers? Are they particles? What's their color? What type of plastic are they? And it's sort of like a forensics, trying to sort of figure out what's the major source. This is just what it looks like under the microscope. So these tiny little particles. And you can see that that little bar there, it's hard to see, but that's 20 microns. So these would be maybe 100 microns in size, right? Very, very, very small. And we shoot it with the laser, and it gives us a spectra, and it tells us, what type of plastic it is. And so what we found is that, 71% of the mussels that we sampled, and again, this was a small sample size. This was just an initial study. But 71% of the mussels we sampled had microplastics in them, and they had anywhere from 1 to 7 microplastics, per mussel. So they're there. But the big question is, what is 1 to 7 microplastics per mussel? What does that really mean in terms of an impact on the health of the organism? So we expanded our study to a much greater area of the Grand River watershed. And we looked at not only, we looked at multiple species within it, and we also improved our ability to detect microplastics in tissue over time. And so we also focused in on some particular wastewater treatment facilities. This is one in Cambridge called Hespeler. So we actually sampled organisms, like we know where the outfall is into the river, into the Speed River. So we sampled upstream and sampled mussels downstream, and then sampled mussels far afield, much further downstream, just to look at, to see whether, proximity to the outfall, whether that has an impact on, because we know with other contaminants it will, pharmaceuticals, perfluorinated chemicals, right? Your position relative to the outfall will impact. But we wanted to see if that was true with microplastics. And again, we go through the same thing. There's Emily happily dissecting mussels. She looks so very excited to be doing what she's doing. But then we're isolating them, digesting away the tissue, looking under the microscope, doing spectroscopy to confirm, it's actually plastic. And we looked at larger mussels, so those native mussels, but we also looked, you may have seen these, they're called fingernail clams or pea clams. They're really, really tiny. You'll find them everywhere. And so, yeah, their abundance is very useful because we can find them in a lot of different locations. But they're really small, called spherids. These are some of the particles, just to give you an idea of what we're finding in them. So you can see there's a variety of different types: fibers, fiber bundles, fragments, films. You can actually get these perfect little spheres. That's the kind of stuff you would find in personal care products. Micro beads, they call them sometimes. And then sometimes we find pieces of foam, right? Anything that's, you can imagine is in commerce, we'll find in them. So the big question was the amount of microplastics upstream and downstream, the hypothesis being downstream, you'd find more microplastics downstream of the wastewater treatment facility. But what we actually found, and I'm not going to get into this too much, but you can see there's not a lot of difference, right? The lighter color bars are upstream and the darkest bar is downstream. And so you can see there's really not much of a difference. We're definitely detecting, about 20 particles per 60 grams. So let's say 20 particles per mussel. But we're really not seeing a difference upstream or downstream from the wastewater treatment facility, which is sort of interesting. This is kind of what we're finding. So this is morphology, and basically we're looking at, what are the types of particles we're finding most of. So the white bar is fibers. So you can see that the particles that we're finding in these mussels is dominated by fibers, right? Which again is interesting. And then we also look at things like color, just again trying to source it. People like to record color of things. One other thing we can measure. And you can see that the sort of light blue, that's clear. So we find a lot of clear blue fibers. Those are the dominant things we're seeing. This is from the digestive gland. So we actually take, the large mussels, we can basically take out their stomach. And that gives us an idea of what they're actually eating. Because just because a microplastic is in the mussel doesn't mean they've actually consumed it. And so, again, you can see we're finding particles. But again, we're not seeing a difference between upstream and downstream of the wastewater treatment plant. So we've gone on. So, I mean, the point of this is that, these mussels, as well as other vertebrates that are living, and the fish as well. Fish, we didn't use fish because they move around, so it's harder, their location is harder to pin down. But you will find microplastics in the stomach of fish, and other, critters that are living in the river. But the big question is that, what impact could that potentially be having on them? I mean, mussels, you can imagine, you're filtering overlying water. You're taking in all kinds of things that wouldn't be nutritious, right? They eat algae and that sort of thing, bacteria. But there's going to be suspended particles and other things that, they haven't gotten this, they haven't evolved and gotten this far without being able to filter out the stuff that's, yummy and the stuff that isn't, right? And so the question is that they're being exposed to these particles, but does it have an impact on their health? So we looked at a variety of different species. So freshwater mussels, we looked at 2 species, but then we also started to look at other things like eligocate worms that live in the sediment. So they're just eating sediment their whole life. Emergent insects, so those are mayflies. I don't know if this works. Kind of, well, not really. But the emergent insects, so they're interesting because they live most of their life in the river and the stream, and then they emerge to mate and then lay their eggs back in the river. But it's interesting for them because when they emerge, you get these mass emergence. They're really important for bats, for birds, for spiders, right? So the question is are they transferring microplastics that would have been in the river, the stream, or the lake to these other, we call it trophic transfer. Are they being transferred to predators? And then we looked at also snails, because snails are not picky. They're just like constant eating machine. Like if you ever watch them, they're just constantly eating. So, what's going on there? Now, just a background, an interesting thing about freshwater mussels, again, not zebras and not quaggas. Those are invasive, not native. But our native species have a really interesting life cycle. They actually spend the first couple months of their life actually as a parasite on fish. So they'll live on the gills of the fish. It doesn't really hurt the fish, but they'll insist on the gills and live off the fish and go through what we call metamorphosis. And then they'll drop off to the bottom of the river, the stream, or the lake, and they'll live the rest of their life there, growing into an adult. So anyway, the point is that they have a lot of different sort of distinct life stages. So maybe the adults wouldn't be impacted by a few microplastics, but what about the earlier life stages, right? The juveniles. Juveniles can be a centimeter in size, or larvae, which are even smaller. So we looked at the freshwater mussels, but we looked at also different life stages, right? The small ones, the tiny ones. They could be potentially more impacted by ingestion of a microplastic than the larger adults. So we looked at also a variety of different types of microplastic. So these are the ones we looked at: polystyrene spheres, polyethylene, because that's one theory, is maybe it's the type of plastic, depends on the toxicity. We looked at fibers, so we made our own fibers, microfibers, and then we used cellulose acetate spheres because those can somewhat break down. And we also looked at different size, because size is also a question, right? Are they more toxic if they're smaller, right? Some particles they'll be able to ingest because they're small, others they won't. And so we looked at different sizes, anywhere from 10 up to 1,000 microns in size. So 1,000 microns would be a millimeter, right? You could see that with your eye. Now the good news is basically we saw nothing happen. So we exposed these larval mussels to up to, right, this is 300,000 microplastics per liter. That's what's in the water, right? So that's orders of magnitude above, because ultimately when we do toxicity tests, we're trying to elicit an effect, right? Because we want to know where do effects start. And so we're thinking, well, 300,000, that's way above, but we might see something happen. And basically we saw nothing happen. You can see here, like, all the viability doesn't change depending on the amount of microplastics you have. But you can see they ingest them right up in the top corner. You can see the spheres of the microfibers, the tiny little mussels. They do ingest them, but it doesn't seem to impact their survival. And then we did it with adults and juveniles, and to try to even further increase their chances of ingesting them, we put it in their food. We feed them an algae mix. So we put the microplastics actually in their food because we know they'll start filtering more when they sense the food, has entered the test vessel. They'll start to filter more. So we put it in their food. And again, we looked at these different sizes, even as small as 6 micron, which is, very tiny. They definitely would be ingesting particles that small. And we went up to 150 million particles per liter in their food, right? So that's like incredibly high. I mean, we're talking about 1 million times greater than what's been measured in rivers and streams. They're fed this food twice a day. And so we looked at, did it affect survival? Did it affect their ability to burrow into the, behavioral endpoints? But then we also opened them up and counted the microplastics that were in them. So this is the effects, right? Percent survival, percent burial. And again, you can see the same thing. It really doesn't have an adverse effect. I mean, up to 150 million particles per liter, it's not affecting the juveniles. So that's a good news story. And these are for the different size particles. Basically, there's no trend. But the interesting thing is that this is showing you what was actually inside them, right? So the more microplastics you expose them to, the more you're going to find inside them. And the blue is when we take the mussel right out of the beaker, right? And we look at the microplastics in them. And the yellow is when we give them 7 hours we call it depuration. So we give them 7 hours in clean water to see whether they can, excrete some of those microplastics. And so even in that 7-hour period, we see a drop. So from the blue down to the yellow, they're able to filter out or excrete a large number of those microplastics. Now the one on the left is showing you the spheres, right? So these tiny little spheres, but the one on the right is showing you fibers. And so clearly the fibers they're not ingesting them to the same extent. It would be like trying to eat spaghetti, you know what I mean, through a tiny little hole. Like mussels, the 6 micron particles could get inhaled in, but these tiny little pieces of microfiber like the spaghetti is much more difficult. So less is getting into them. Now we've gone on to look at, like I mentioned, the Alegachids. So the Alegachids, like earthworms, but they live in sediment, like in the bottom of rivers. They just plow through the sediment their whole life, just sort of ingesting. They get bacteria and organic matter. That's where they get their food from, just like an earthworm would. Emergent insects, they live in the bottom, but they filter water from the river and catch little particles and eat them. And then the snails, again, like I said, they're just machines. So we were looking at survival, but also egg production, so endpoints for reproduction. Because again, there's a theory like, well, maybe it doesn't kill them, but it inhibits their ability to grow or their ability to reproduce. Maybe there's subtle effects. One of the theories on how microplastics impact organisms is called food dilution. So if they're eating a bunch of microplastics that really aren't nutritious, so they might be excreting them, pooping them out, but because it's taking space for normal food, there would be, a lack or reduction in nutrition. So these are some of the theories on how they might have an impact. And basically, again, we looked at a bunch of different sizes, 5 micron all the way up to 100. We also looked at pristine plastic. So if I put like nice clean plastic in, and if I take plastic that I've sort of left out in the environment for a while, because bacteria and biofilm grows on it, maybe we can trick them into ingesting it, right? Maybe they don't like the virgin clean plastic, but if we have biofilm on it, maybe it'll seem more appetizing. Um, and we went up again to in the sediment, we're like 1,000 particles per gram of sediment, 1 million particles per liter for the emergent insects and the gastropods. And basically, we didn't see any impact on them. I'm not going to show you the graph because it's very boring. It's not very exciting. Basically, we haven't seen any impacts. And so, I mean, this is a good news story. And we're going on to look at also soil invertebrates. So a big source, at least in Ontario, a big source of microplastics to agricultural fields is biosolid amendment. So in Ontario, and I'm not sure in Saskatchewan, but in Ontario, there's a lot of biosolid, right? Biosolid being solid sewage waste. That's most of you are better experts at that than me, but has been processed and then it's land applied on agricultural, right? Provides nutrients, organic matter. But there's again a concern that microplastics, as well as other contaminants, are being introduced to the agricultural fields. And what impact does it have on the living things there? So we've gone on to look at, earthworms and springtails. They're little critters that live in the soil. They're detritivores. They break down the organic matter. Isopods, like I mentioned. We've also looked at ants, right? If the ants are in soil that's contaminated, what do they do with the microplastics? When they're building nests, does it impact their ability, the health of the colony? We've looked at American millipedes, which are another type of, detritivore breaking down. And then we've also looked at plants and whether microplastics inhibit plant growth in some way, because there's some studies in the literature that would indicate that they do. But I don't know, I call BS a little bit on that because I'm trying to understand how microplastics in the soil are going to impact plant growth. I mean, there's all kinds of particles within the soil that are not nutritious, right? Like, I mean, they're tiny little particles of minerals and anyway, so we've looked at that. We've also looked at whether it impacts the relationship between the plant roots and the beneficial arbuscular mycorrhizal fungi. And so thus far, again, there's still work to be done, but thus far we basically haven't seen any impacts on these species. And so again, like it's a good news story. But the problem is that in the literature, there's conflicting evidence, right? You get these sort of one-off papers where they've reported some effects, and that's the one that gets sort of taken up by the media, right? Obviously, you're trying to sell papers or, magazines. And so anyway, our work is trying to sort of, through sound scientific method, really look at the impact of microplastics in the ecosystem. So one of the things that I'm sort of advocating, I mean, I think we can all agree that, plastic or microplastic in the environment is not a positive. I mean, we've got a waste management problem. But what our group is trying to do is, we need to assess the risk of that plastic. Now, plastic bags in the ocean, yes, they're going to impact turtles and whales and albatross, no question. But, what risk do microplastics pose? And the idea is that, well, if there's not a risk, we can sort of let that issue go and move on. That's not the point of it. But the point of it is, that like anything, there's limited resources that we can put towards solving problems. And so should we be looking at, engineering solutions to microplastics in water or soil? I mean, can you imagine what that would look like and how that might be destructive? You can imagine going through a river trying to remove microplastics from sediment. It'd be crazy, the impact you would have on that ecosystem. So the idea is, where should we be, investing, and other academics would kill me for saying this, but should we invest in more research in the impact of microplastics on ecosystems? Some would, say, of course, you've got to give me lots more money to do more experiments. But my argument would be no. We should be investing that money and time in dealing with the issue, whether that's researching plastics that are more readily degraded. Also, there's the sociological element, right, of getting us to use less plastic. We're also horrible at recycling plastics. Does anybody know what percent of like municipal plastic actually gets recycled? That's for some municipality, yeah, 5. I think Guelph does pretty well at 20, right? Which is astounding to me because I put a lot of effort cleaning out my plastic, and then putting it in the right bin. But anyway, so the other issue is that the concern some scientists will bring up, well, look at our production of plastic. It's going up year after year after year. So theoretically, you're going to end up with more and more microplastics in soil, in water. So they're not wrong. But again, where do we invest our resources? More research on the impact of microplastics or engineering to try to remove them from the environment? Or do we invest those limited resources at the sort of upstream, like in terms of the plastics we use, better recycling, and just better waste management? So again, I don't have the answer for you, but these are some of the sort of things that we're thinking about. So anyway, just acknowledge, like, I'm the leader of, so I don't, it's my grad students who do all the work. So thank you to Urena and Quinn and Colleen and Emily. They're the ones who really are the ones who do all the work. I just get to present it. And then Karen Kidd, my collaborator at McMaster. But this research has been funded by Environment and Climate Change Canada, as well as NSERC. Health Canada is involved too, because it's seen as an emerging contaminant, and it's important for federal and provincial governments to get data, because if you're going to properly assess the risk of a contaminant, you need to understand its fate in the environment and the effects that it can have on organisms. So how they're going to use that data in decision making, we'll see. We just generate the data, but yeah. So anyway, the idea here is not to say, hey, don't worry about microplastics, move on with life. I mean, we still have a plastic problem, but maybe the microplastics in the stream or in, the field are not as dire as it may be communicated to us, right? So with that, I will leave it. I have some references here. Not you're going to read those, but if you, just to say what I'm saying is not completely made up, and it's just my opinion.